All life on Earth, be it bacteria, humans, or trees, orient themselves along one fundamental law of physics, and that is gravity. So, for instance, a tree grows its roots into the soil towards gravity, and its branches and fruits up to the sky, up to the light, away from gravity. And it's not by coincidence that I chose this symbol, because it looks a little bit like the Jung-Stiftung symbol. And with that, I'm honored to be opening today's 4th Jung-Symposium with my talk, Brain and Balance. My name is Maximilian Friedrich. You have seen some of my introduction, I think, in that beautiful video. And thank you for giving me the opportunity to have such a video. It's an amazing achievement. So, complex human motor behaviors, such as upright gait or orientation in three-dimensional space, critically rely on our ability to sense our position in relation to gravity. And this is mediated by the vestibular system. For some reason, the vestibular system consists of the inner ear's labyrinth, which encompasses the 3 semicircular canals and the 2 otolith organs, which send their signals through the vestibular nerve into the brain, where they cover major areas of the central nervous system. And you can think of the vestibular system as some sort of your internal 3-dimensional coordinate framework that allows you to coordinate eyes, body movements, and your orientation in 3-dimensional space as a function of gravity. So what happens if it fails? So it follows from the function that vestibular failure causes postural instability, falls, and gait problems. But there's one set of symptoms that we pay particular attention to in the clinics, and those are eye movements. So here I brought you 2 patient examples, 2 cases of patients who presented to RER with acute dizziness. So you can see these wiggly eyes, and these eye movements is what we call nystagmus. And nystagmus is an eye movement disorder that tells us a lot about what is going on in the brain. So what happens next? If we see and clinically judge these eye movement disorders, we mostly order neuroimaging. And as you can see in these 2 cases, that was a very good decision, because we see massive problems here. We see a cerebellar stroke or a thalamic hemorrhage. So what we do next as clinicians, we use this information from imaging, the lesions, and we try to register that lesion in our head to our knowledge of brain anatomy. For some reason, my text is gone from these slides. I'm sorry. So I just need to walk you through more globally. So why do vestibular disorders matter for you and for us? Well, there's a 35% lifetime prevalence of vestibular disorders globally, and also over 80% of people over the age of 80 report balance trouble. Also, balance issues lead to falls, anxiety, and ultimately they amount for a significant part of morbidity in neurological disorders. And also we look at an economic problem, because over $8 billion are spent every year for emergency care of dizzy patients. So, vestibular disorders matter, but we face significant challenges. And one of these challenges is that we have a striking lack of neuroimaging resources that help us formalize and systematically investigate the relationship of structure and function when we see a stroke lesion. And the structure-function relationships of vestibular brain networks are still poorly understood. So this is an anatomical or structural dimension of the problem. On the functional side, vestibular symptoms such as nystagmus are very hard to measure objectively. So this all boils down into one big problem, that is, we have hardly any effective treatments. And the same was true for quite a while for other neurological disorders, such as Parkinson's disease. But with the advent of neurostimulation methods, such as deep brain stimulation, we can now offer patients with severe neurological disorders hope. And what happens is that a neurosurgeon in an intervention will insert, for example, an electrode into the deep structures of the brain, and this electrode is hooked to an impulse generator which delivers electrical current into these brain regions. You can think of this as some sort of a brain pacemaker. And if we do studies and research in these patients, so here you can see how that looks like. So we reconstruct the electrodes of several patients and we visualize that in a common imaging space here. This is a model. And you can see that all of these electrodes, they seem to be targeted to one spot, and they are. They are targeted to that orange spot, the subthalamic nucleus. And if I give you that scale bar, you can see that we are looking at a structure that is really only 1 centimeter. So millimetric precision matters and actually decides between success and failure of these interventions. So in finding neuromodulation targets and optimizing treatments, there's a crucial need to have a good understanding of the anatomy and how it relates to the function, and the precise mapping of both is crucial for success. Unfortunately, vestibular patients with balance disorders today, they really do not benefit from these massive advancements in neurotechnology. And I want to see if we can change that. So starting off with anatomy. How can we causally map brain functions? And I need to give you a little historical throwback with maybe one of the most prominent examples of a lesion case in history. And this is the patient Phineas Gage. He was a railroad worker in the 19th century in America, and he suffered a terrible accident, an explosion that drove a metal rod through his eye and through his skull and damaging his frontal part of the brain. And believe it or not, he woke up and he was fairly normal, but like neuropsychological testing yielded very circumscribed deficits. And these deficits, when mapped, really told us how the frontal brain actually works just by mapping the problems when it's not working anymore. So this was when the lesion study was born. And today we have neuroimaging. So that means we can have a map several lesions from several patients with the same problem. And we can look for the spot that is the common denominator of all these lesions. And we think of that as the common causal substrate of the function we're investigating. So let's see how it holds up in our example, coming back to our patients. So one patient had a lesion down here in the cerebellum, the other one up here in the thalamus. Well, that didn't prove the point. It doesn't work. And I need to give you some more perspective in history. On the left, you can see Konstantin von Monachov, the head of neuroanatomy in Zurich in the 19th century. And he had a very influential concept of diaskesis. Diaskesis basically means separation. And he posited that not single brain regions, but in fact networks of different brain regions that are connected through white matter pathways subserve neurological functions. So now it makes more sense. I highlighted you on the right in that little circuit model all the white matter pathways that are related to vestibular function. And now it becomes clear that lesions in many spots of the brain can cause vestibular problems, but don't overlap in one single spot. So brain functions are mediated by networks of brain regions. So let's dive in. How do we map vestibular functions and vestibular regions through lesions? And here I brought you one case that I saw together with Laudin Schappe from the University Hospital of the Saarland. And this is a patient who presented with a very strange and rare eye movement disorder and imbalance. So it looks subtle, but what you can see is that his eyes move upward and towards the nose, as if he was focusing something that comes close to him, which was not the case. So let's go back to our vestibular coordinate framework that I showed you initially. The problem or imbalance that we're looking at really does not relate to rotation, but to translation. And translation is a movement that happens when we walk, for example, because our head in this movement is not rotating, but it's moving back and forth on the backward-forward plane, and this is what we call a translation. And interestingly, translation has never been mapped in the brain. So, we looked at an MRI scan and we saw, yes, the patient had a lesion, a tiny one in fact. And we used neuroanatomical inference to map typical vestibular brain regions in the midbrain, here in green and blue, the INC and the RIMLF. We will talk more about the function later. And here in red, you can see the lesion. And we have transferred these data into a high-resolution cytoarchitectonic MRI atlas. And we saw, well, the lesion does not really overlap any of these two regions. So we're looking at something else. So what we did next we leveraged histological data that is co-registered into the MNI or MRI space, so we can really side-by-side compare histology and neuroimaging. And you see that the lesion falls in a spot in here that is very close to the region called the posterior commissure, a white matter pathway, but also to nuclear structures. We were hooked and very interested, so we collected 66 lesions that happened in that midbrain area of this single patient in a follow-up study. And of these 66, 44 had the same problem, whereas 22 served as our controls and did not have the same eye movement or vestibular problem than our patient. And we did voxel-wise statistical mapping, and we found that this tiny little spot in the midbrain was statistically highly significantly associated with the eye movement disorder of interest. So we were wondering, what does this spot do? It was really not covered in most of our anatomical atlases. It was a true terra incognita. So we wondered, can we get our hands on more causal sources of information? And we thought of DBS, deep brain stimulation electrodes. And in fact, we found one patient, serendipitously, who had a side effect from his deep brain stimulation that looked just like the problem the patient had. So we modeled the electrode's electrical field in the patient's brain, here in blue, and we also mapped the spot that we found using statistics from the lesions that we think is involved in this vestibular function. And here in blue you can see the electrical current that did not cause the problem. It did not really overlap this region. However, the electrical field that did cause the problem, the eye movement problem in this unrelated patient, really did overlap it in a considerable manner. So, as a preliminary take-home message, vestibular brain maps and brain atlas maps can help us contextualize and understand neuroimaging findings and investigate the function of the brain in unprecedented detail. So how can we map vestibular networks? So for this I need to tell you a little bit about the human connectome. So using advanced neuroimaging methods such as diffusion tractography or functional MRI mapping, we are now able to map the large-scale connectivity of the human brain. And these resources have been made possible by large-scale efforts in several centers in the world, and now we have some sort of a normative wiring diagram of the brain, of a normal brain. And we use this data to infer the effects of lesions, interventions like deep brain stimulation on other regions of the brain. And this method, termed lesion network mapping, has been made possible and in fact invented by my mentor and PI, Michael Fox, at the Brigham. And shortly thereafter, Andy Horn, who is also at the Brigham and Women's, has built a whole new branch on the deep brain stimulation mapping using the connectome. So now we can test von Monakoff's theories in real life. And here I show you another case that we published recently. This was a patient who underwent deep brain stimulation for Parkinson's disease, and he said, well, after the deep brain stimulation, I started seeing towers and poles tilted like the tower or the Leaning Tower of Pisa. And I thought, that doesn't make sense. That shouldn't be. That is a side effect, and that sounds vestibular. So here's a video. In this video, I test the electrode contact that causes the problem, and you can see that increasing the current really makes his eyes go away from one another vertically and his head tilted to the side. And you can think that in this patient we really messed with verticality perception. And now if we tone down the deep brain stimulation, it's gone. It's almost gone. And if we turn it up again, you can see the eye moving up, moving up, moving up, and he said, wow, this is very uncomfortable. I feel like I'm falling to the side. So coming back to our vestibular coordinate framework, this is a role-playing problem. So this is about the vestibular sense of our orientation to the gravity vector. And for this, we have a very good idea where it should live in the brain, and this is the nucleus of Cajal. So we know that this nuclear area, a tiny little nucleus in the midbrain, is involved in verticality perception and head position and verticality. And we said, well, we guess the deep brain stimulation electrical field will hit that target. And this is precisely not what was the case. Here in dark red, you can see the electrical field that causes the problem, and it was very far away. And here you can see in this dashed line. So we thought, well, if we're not hitting that spot, we need to hit a fiber pathway maybe that connects the spot to other spots in the brain. And we used the connectome that I showed you to map that. And in fact, in the healthy human brain, there is one tract connecting the nucleus of Cajal and other areas in the midbrain. And this is exactly the tract that is being hit by this electrical field in that patient. So what we did is we said, well, we're not sure if we believe that. We programmed the deep brain stimulation electrode electrical field away from this tract. And in fact, it was gone. The symptom was gone. And the patient still had a very good response in terms of his Parkinson's disease. So we were wondering, is that tract true? Is it a true thing? And we went down into history and we found yes. Only in cats and in monkeys, this tract connecting the nucleus of Cajal and the zona incerta in the midbrain has been described, but not in humans. And in fact, if you stimulate this tract in the very same spot then as the patient in a cat, the cat's head tilts to the side and the eyes move vertically from one another, just what we saw in our patient. So this goes to show and to prove that we can stimulate and that we can modulate vestibular functions in a precise and almost surgical way if we know where and if we know what. So let's move on to another recent study. In this study, we were more interested in the higher order organization of the cortical brain networks that give us our sense of orientation in space. So coming back to our vestibular coordinate framework, we were now looking not at single planes of that framework, but we were really looking at the complex three-dimensional representation of myself in space. Where am I in space? Those are rare cases, but we managed to find 23 lesions outside of the brainstem that did cause vestibular sensations related to orientation and position. And again, no significant overlap. So you can see these lesions happen in many, many different brain regions. Well, so we used the connectome, this time the functional connectome, to understand for each individual of the lesions what other brain regions these are connected to. And what we found was striking. 96% of the lesions were now connected to the same spot in the insula, whereas almost no lesion overlapped. Now we have a common network that they align with. And this made sense. If we go back in history once again, Wilder Penfield, the father of modern neurological mapping of brain function with electrical currents, has predicted that around the insula, around that temporal side of the brain, here denoted with Ls, there must be a place, or there must be cells that give us our sense of orientation, vestibular orientation in space. So we teamed up with Liang-Kun Ren from Beijing, who had access to cortical stimulation data of epilepsy or of patients undergoing epilepsy surgery. And we really wanted to put our network to the test. So we hypothesized if our lesion network was really showing us what we thought, a sense of vestibular orientation in the cortex, the brain stimulation spots should align. And this is precisely what we found. So here in yellow you can see the brain network associated with lesions causing vestibular orientation problems. And in red you can see the tiny little electrode spots that in individual patients, when stimulated, cause the same problem. And the red ones align very well, whereas the blue ones in here, those were the electrode spots that specifically did not cause vestibular problems, and they fell outside of this network. So the conclusion here is that the connectome, with all its assumptions, can help us identify targets for vestibular neuromodulation and potentially also for therapeutic modulation of function. So let's come to the other part of the story, the functional mapping. How can we map vestibular functions? So as you might have seen, we clinicians love to take videos of patients. We love to take arm movement videos because we use these videos and watch them forward, backward, slow, fast. We discuss them with colleagues because it's complex. So I thought, how cool would it be if I had a means to use this information from the videos to really get quantitative information out of that? So I devised a study in which we presented a moving stimulus on a smartphone, a normal smartphone, and we videotaped the eyes of the subjects in this regard watching that stimulus, okay? And that stimulus created a nystagmus because it was like a rolling stimulus. We call that optokinetic band. And then we developed a deep learning algorithm, a so-called visual perceptive convolutional neural network that allows us to track key points in a video, and in this regard, key points around the eyes. And we use the time series of these key points to track the eyes and now get quantitative information. So, of course, I was suspicious. I thought, is that really working? And we put the method to the test. We compared it with the current gold standard, video oculography. This is a highly specialized goggle that you can only find in very few centers worldwide. It's orders of magnitude more expensive than your smartphones, and it does the job. So here you can see the traces that are being produced by both methods. Top row is the smartphone approach in green, and the bottom row is the gold standard. So it looks decent. It may not be as detailed, but it's still decent. But now, within clinically derived boundaries of equivalence, we found that our approach using smartphones can extract kinematic characteristics of nystagmus with equivalent accuracy than this method. So now we have a means in our pockets to really measure eye movements. And coming back to our patients, here you can see these two examples were not in the training data set. So those are unseen examples. And the algorithm works very well. The eyes are tracked nicely. And now we can use these videos that we have everywhere in our hospitals, on our smartphones. We have video databases everywhere to really extract digital biomarkers of vestibular symptoms or of neurological symptoms more generally. And now let's come back to the mystery spot that kept me up for several nights in the last years. So up here, I show you again one example lesion. And interestingly, in these 66 cases that I told you about initially, 17 had videos available. So in this subgroup, I really wanted to know, how does this nystagmus exactly behave? And here you can see a trace extracted from the eye movements of the patients. These are convergent and divergent movements happening at a frequency of around 2.2 hertz. And now I teamed up with non-human primate researchers from Houston, Texas, with Balab Das's group. And we just so happened to find a couple of lesion cases in monkeys. And these lesions were made by pharmacological precision injections that deactivate neurons in that area. And you can see that here, the monkey brain, and here the human brain, those are the same spots. And interestingly, the eye movement consequences were also almost identical. So you could create the same problem as in our patients in the monkey, happening at a very similar frequency. So my take-home message for you is that I believe precise mapping of anatomy and function and their interrelation at least gives us the groundwork, or lets us lay the groundwork for precise and causal mapping of brain function, which we need to modulate and modify and hopefully improve brain function for patients with vestibular disorders in the future. And with this, I want to thank my amazing team of mentors, Michael Fox, Andreas Zweigal, David C., and Jens Volkmann, for helping me along the way, for inspiring me. And last but not least, I want to thank the Jung-Stiftung für Wissenschaft und Forschung for trusting me with this project. It comes timely, it's just the right time for me now, and I'm really looking forward to working with you and to making this project happen. Thank you for your attention.